Hi everybody, welcome back to Brain and Behaviour Teaching Block 10, the Principles of Neural Representation. My name's Tom Hartley, and in the last lecture of the series today we'll be talking about principles that might underpin functional anatomy and topographical organisation in the brain. We know that different parts of the brain do different things, but are there any patterns, are there any principles that underpin the determination of which part of the brain ends up doing what function? That's what we're going to be looking at today. Over the last 3 weeks, we've gradually been zooming out. We began by looking at perhaps the very smallest units of cognition, the individual neurons. And then in week 2, we began to look at how patterns of activity across groups of neurons or populations could represent meaningful information. In week 3, we began to look at how different populations communicate with one another and how those connections between populations of neurons can affect systematic transformations from one pattern of activity to another pattern of activity so that the brain can do something useful. So this week we want to see whether we can identify general principles that underpin the organisation of function in the brain. As I say, we know that different parts of the brain do different things, but when we look carefully, we tend to find that nearby parts of the brain, especially the cortex, tend to do similar things. And that leads to the idea of cortical maps, where we see the function of different neurons arranged systematically on the surface of the brain. And those maps have some interesting and characteristic properties wherever we look in the brain. Parts of the map that require more detailed processing are often accorded more space in the brain, and we see some signs that these maps may be plastic. They may be able to change their shape and structure over time to accommodate changes in our experiences or our sensations. Later on, towards the end of this session, I'll talk about a type of neural network model called a self-organizing map that can explain not just the emergence of maps but some of these properties. One of the intriguing features of topographical organization in the brain is that it seems to take place on every scale, from the individual neurons, as illustrated here in the top left diagram, right up to the size of the entire cortical surface, as shown in the bottom row of images. And later on in this session, I'll be taking you through some of these patterns of organization at different scales. And we're going to begin our discussion with vision, because vision is probably the best studied of all of the processes in the brain in terms of the topographical organization. You'll remember, going back to week 1, we talked a lot about single unit recording of visual neurons, simple cells in primary visual cortex, and their characteristic tuning to different stimulus orientations. And I showed you that video of Hubel and Wiesel recording the activity of an individual cortical cell. It turns out that when you look at these simple cells in visual cortex, you find that they're tuned to multiple dimensions of the visual stimulus, as you'd expect, because this is where all of the visual information for the brain is arriving. So all of that information has to be carried in some form by the neurons at that staging post. And so we find that neurons, individual neurons, are tuned to a certain retinal location, so a certain part of the visual field, a certain orientation of the stimulus, as we saw before. But they're also tuned to respond to ocular dominance. In other words, some neurons respond more strongly to stimuli presented to the left eye, some respond more strongly to stimuli presented to the right eye. They're also tuned to the direction of motion and to different colours. So how is all of this information, how are these different tunings organised on the surface of the cortex? Is there any pattern, or are those neurons just arranged randomly in the primary visual cortex? One of the things that Hubel and Wiesel discovered in their early work was that there did seem to be some kind of pattern. So you'll recall that they were recording from individual cells on a recording electrode, and over successive sessions, over different days, the electrode tip would advance through the brain, so it would go deeper and deeper into the brain. And what they observed, and you can see this plotted in the diagram on the left hand side here, is that as their electrode advanced through the cortex, they found that the ocular dominance tuning, represented by black or white circles here, and importantly the orientation tuning of the neurons that they discovered, was gradually and systematically changing as the electrode moved through the brain. So you can see that at the very top of the diagram, the neurons that they're recording from are responding to an orientation of about 30 degrees with respect to the vertical. A little bit further on, they're responding to a vertical orientation, and then the orientation is systematically changing as they advance the electrode through the brain. And the idea that Hubel and Wiesel came up with to explain this was a pattern that's shown on the diagram on the top right there. So they suggested that orientation columns in the visual cortex, in the primary visual cortex, were arranged systematically so that for a particular ocular dominance, left or right, you would see a row of columns so that the orientation gradually tilts through space. And what they were seeing was, as the electrode went through, they would be successively sampling from different columns in this structure. In the end, it's turned out that their model, although it was broadly correct, there is some kind of patterning to the orientation tuning. It's a little bit more complicated than that, and that's what's illustrated in the diagram at the bottom right of the slide here. It's a little bit hard to follow this, but I'll be explaining this patterning a bit more on the following slides. But if you look at the left-hand side, the black and white part here, you can see that you've got these stripes running vertically, separated by thick black lines, and they correspond to, each stripe corresponds to an ocular dominance channel, so left or right favouring ocular dominance. And we're looking down at the surface of the cortex, and what you can see is those contours, those lines, represent the orientation tuning of different patches of cortex. So each line shows you the direction that was favoured by the neurons in that area, and you can see points around which all the possible orientations are organised, so-called pinwheels. I'm going to explain those a little bit more and perhaps more clearly in some of the next slides, so don't worry if it's not too clear from that diagram. What you can see from the diagram on the bottom right of this thing, the coloured in version, is that it's subsequently been discovered that the colour tuning of the neurons is also arranged around the same patterning, the same scale, so that around the location where those pinwheels occur, where all the orientations come together, there tend to be more blue sensitive neurons, whereas where the different oblique orientations are represented tend to have red and green tunings. The key thing to notice at this stage is that all of these different dimensions are organised systematically on the surface of the cortex at roughly the same scale. We can see ocular dominance, we can see orientation, and we can see some organisation of colour. So what about retinotopy? What about the organisation of visual field tunings among these neurons in visual cortex? Are they arranged in some systematic way? That can be very difficult to discover using the single unit techniques that Hubel and Wiesel adopted, because it seems to be organised on a much larger scale. The hint to this comes from the tunings of cells recorded from the same electrode. So what's shown here is a number of different receptive fields from neurons recorded from the same electrode in monkey striate cortex. And what you can see is that they're all located in roughly the same region of the visual field. So the arrow there points towards the centre of the visual field. Each of these cells responds maximally to a small region of the retinal image, roughly the same location. But trying to understand how this was organised would be almost impossible with single unit techniques. As the electrode moves, you don't see very much change in the tuning. In retrospect, it's remarkable that they discovered anything at all about the functional organisation of visual cortex using single unit techniques, because looking at the function of individual brain cells one at a time is not the best way of revealing the overarching pattern. In fact, Hubel himself likened it to cutting the lawn with nail scissors. Other neuroscientific techniques may be much better suited to revealing structure on larger scales, and that's what we'll be looking at in the next few slides. One example is optical imaging, which has been very important in revealing those pinwheel structures that I discussed earlier on. Optical imaging works by recording the visual appearance of the surface of the brain and looking for signals that are associated with neural activity. In its simplest form, this can be intrinsic optical imaging, which looks at the rapid changes in appearance that are associated with different degrees of blood oxygenation. But more advanced methods use voltage-sensitive dyes to detect cellular changes that are associated with the electrical activity of the neuron, and these are capable of imaging individual brain cells and their activity. So optical imaging can give us a larger scale and two-dimensional view of the organisation of cortex. In case you weren't familiar with the term, and I wasn't, that's a pinwheel shown on the left-hand side there. It's a kind of windmill toy that you might get at a fairground or something like that. And you can see how its structure resembles the orientation tuning of neurons in a patch of cortex on the right-hand side. So here the different colours are being used to represent the different tunings of the neurons in this part of the cortex, and you can see that all possible orientation tunings are arranged around a single point in the middle. So that's what's meant by the pinwheel. So that was some of the optical imaging data that first helped to describe this pinwheel-like structure in the orientation tuning of visual cortex. And what you can see below in the slide is some data from a more recent study that used more advanced methods and looked at a larger patch of cortex. We'll be talking about this study in a bit more detail in just a moment, but the thing to notice first of all is the scale at which this occurs. So you can see within this patch of cortex that's approximately 2 millimeters across, there are several pinwheel structures represented, and that means that all of the possible orientation tunings are represented multiple times within that relatively small patch of the cortex. What you're seeing at the moment is a video from a more recent study by Oki and colleagues in 2006 that uses the more advanced techniques that can resolve the firing of individual brain cells, and that's what you're seeing here. On the right hand side is the stimulus that the monkey is viewing, and you can see that each orientation of stripes is being presented for a short period of time with a grey screen in between. Each of those bright dots is an individual brain cell responding to the stimulus, and as we progress through the different orientations of line, you can see that patches of the cortex are becoming active in turn, and we're moving kind of anti-clockwise around the centre of the image that you're seeing. So what you can see on this slide is a summary of that data. So on the left hand side we've got a picture of the patch of cortex with each of the bright spots, each of the neurons being coloured in according to its orientation tuning. And this figure highlights a few of those neurons with numbers 1, 2, 3, 4, 5, 6. So if we look at the data from individual neurons, so number 1 is shown in figure B at the top, you can see that what you're looking at there is the response of that particular neuron to each of the different orientation stimuli, and it comes to a sort of bell-shaped curve that peaks at a specific point, a particular orientation which that neuron prefers. Same with number 2 just below, number 3 just below that. Each of the neurons has its own preferred orientation, and so those are the things that are being plotted in the pinwheel diagram to the left. And the important point for you to take away from this is that even if we go down to the very finest level of organisation in the brain, looking at the individual brain cells, we find that nearby neurons do similar things. So optical imaging seems really well suited to picking up the organisation of cortical function on this scale between sort of 100 micrometers and a couple of millimeters. But all of the neurons that you just saw are most likely responding to a very small part of the visual field. And we know that because retinotopic organisation, the mapping between the retinal image and the visual cortex, is on a much larger scale. And how do we know that? What kinds of techniques can we use to pick up organisation on that kind of larger scale, millimetres to centimetres? And perhaps surprisingly, some of the first evidence came from analysis of human brain injuries. In the early years of the 20th century, Inouye studied the brains of soldiers who'd been affected by gunshot wounds, and because of the development of new high-velocity sniper rifles, these bullets didn't always kill the person who was hit by them, but sometimes would pass right through parts of the head, right through parts of the brain, and cause very specific brain injuries that left them alive. And if these injuries affected the back of the brain, the occipital lobe, the patient would very often be left with regions of blindness, parts of the visual field that they could no longer see. And Inouye and later other scientists began to analyse the relationship between the parts of the brain that had been damaged and the regions of the visual field that had been affected. So what you're seeing now is a remarkably detailed diagram that was constructed by Gordon Holmes and other scientists working using similar methods mainly during World War I. And what you can see on the left hand side of the screen is a depiction of the posterior part of the brain and its medial surface. You're looking at the calcarine fissure of the occipital lobe where primary visual cortex is, and there are different kinds of shading that have been applied to different parts of primary visual cortex. And on the right hand side of the screen you can see a depiction of the visual field and the parts of the visual field that have been affected by the different brain injuries. The first thing to note is that of course we're seeing the left hemisphere of the brain but the right visual hemifield, because the left hemisphere of the brain receives visual input from both eyes but always from the right hand side of the visual field. Another thing to notice, if you look very closely at the shading in this image, is that regions of the visual field above the horizontal meridian are represented in the lower part of the brain, so that the map in the brain is flipped upside down compared to the outside world. A final thing to notice is that the very small part of the centre of the visual field is represented by a very large region at the back of the brain at the occipital pole. The part of the image that we focus on, that we attend to, gets a lot of space at the back of the brain, whereas the outside part of the visual field is represented by a relatively smaller part of the brain, much deeper inside. So historically we were able to get quite a detailed understanding of the retinotopic organisation of visual cortex from this intensive study of patients, but nowadays we have techniques that would allow us to get the same kind of information much more quickly and without anyone getting hurt. And in particular, functional magnetic resonance imaging is a fantastic tool for investigating functional organisation on this millimetre to centimetre scale. While the resolution of fMRI isn't fine enough to pick up on structures at the scale of pinwheels, so less than a millimetre, at larger scales it's perfect. It's non-invasive, so it's applicable to healthy humans, and we can capture data from the whole brain at the same time, and that compares very favourably to single unit recording, but also to other techniques like neuropsychology, brain stimulation, or even optical imaging, where we're limited to looking at relatively small parts of the brain at any one time. What you're seeing at the moment on the screen is an expanding ring, a checkerboard pattern that's stimulating your visual cortex. And if you keep your eyes focused at the centre of the display, what should be happening is that in your cortex we should see a wave of activity starting at the back of the brain, the occipital pole, when the checkerboard's in the middle of the screen, and gradually moving forwards in the brain as it expands towards the edge of your visual field. With fMRI we can track those waves as they move through the brain, and we can work out when each part of the cortex becomes active. And so we can produce this kind of colour-coded map which represents which part of the cortex is active at each stage of that stimulus cycle. So here in the bottom left-hand part of the image you can see the responses of different parts of the occipital lobe to the expanding ring stimulus, to the eccentricity of the stimulus, and on the right-hand side you can see how the response tracks a rotating wedge stimulus. So here we've got this wedge-shaped checkerboard that's rotating downwards from the top of the visual field down towards the bottom of the visual field, and you can see that that's associated with a wave of activation that moves from the bottom part of the occipital cortex up towards the top of the occipital lobe. In this video we focused on vision, and we've seen that functional organisation is structured on every scale from the single unit right up to the entire occipital lobe. And we've seen that some methods are better suited to investigating organisation on different scales. But we've also seen a pattern emerging. Nearby parts of the brain seem to do similar things, and that's true whether we look at the individual units in a pinwheel or whether we look at the entire surface of the retinotopic map in visual cortex. So in the next video we want to see whether this is a general principle. Does it apply to other sensory modalities? Does it apply to movement and action? Does it apply to even cognitive types of representation?